last lecture we had completed chapter respiration and circulation. Here we start new chapter, inheritance and variation. It is the third chapter, inheritance and variation. You know about Mendel, Gregor John Mendel. It is the father of genetics. He published his work on inheritance of traits. Mendel published his work on inheritance of traits. Inheritance means passing of character from one generation to next generation. In 1866, he published his work in 1866, and up to 1900 it is not recognized. Why it is not recognized? Because that time there is no communication like recent time, and his work was not recognized and not accepted by the other biologists. Why his work is not accepted by other biologists? John Gregor Mendel, his approach of using mathematics and statistics for explaining biological concepts, it is or phenomenon, it is not accepted by other biologists. Other thing, as continuous variation takes place in nature, variation means what? Changes. Number of changes continuously changes in the environment. There is a variation takes place in the environment, and the concept of Mendel is the factor or genes. That time Mendel's called factor to the genes. This concept, factor is stable and discrete unit. This gene or factor is a stable and discrete unit. Stable, which is not changed, and discrete means it is distinct and individual. But there is a changes takes place continuously. म्हणजे हे जे रिलेशन इथे ट्रू होत नाही, सारखं एन्व्हायरमेंट मध्ये बदल होत आहे. आणि हा मेंडल्स तर सांगताय की कसा आहे जीन स्टेबल अँड टू डिस्क्रीट आहे. मग ही कन्सेप्ट पण बाकीच्या बायोलॉजिस्टला ॲक्सेप्ट रिकॉग्नाइज झाली नाही, म्हणून त्यांनी ते ॲक्सेप्ट केली नाही. थर्ड पॉइंट आहे पेअर ऑफ अलील डिड नॉट ब्लेंड इच ऑल. जी पेअर असते अलीलची, फॉर एक्झाम्पल कॅपिटल टी और कॅपिटल टी स्मॉल टी और स्मॉल टी आपण पाहिलेले तुम्ही चॅप्टर मध्ये मागच्या वर्षी मेंडल्स. तर हे डॉर्फ आणि हे स्मॉल टी डॉर्फसाठी, आणि हे कॅपिटल टी स्मॉल टी हे टॉल हे कॅरेक्टरसाठी. हे दोन जीन्स आहेत, दोन अलील्स आहेत. टी capital T small t alleles are. These alleles, this pair of allele did not blend each other. म्हणजे एकमेकांवर त्याचा इफेक्ट होत नाही असं सांगतात, परंतु जर इफेक्ट होत नाही तर मग चेंजेस कसे होतात? तर हे बायोलॉजिस्टला ही कन्सेप्ट जी आहे ती ॲक्सेप्ट केली नाही मेंडलची. आणि तिसरा पॉइंट आहे मेंडलचा, का त्याचा जो वर्क आहे तो ॲक्सेप्ट केलेला नाही. व्हाय his work is not accepted by other biologists? Because he did not know the location of factor or genes in the gametic cell. तो सांगू शकला नाही की या फॅक्टर्स किंवा जीन्स लोकेशन काय आहे गॅमेटिक सेल मध्ये, कुठे लोकेट आहेत, कशावर लोकेट आहेत जीन्स, हे त्याला सांगता आलं नाही. तर असे नंबर ऑफ फॅक्टर्स आहेत की जे या मेंडल्सचे बाकीच्या बायोलॉजिस्टने ॲक्सेप्ट केले. देन इन 1900 there is a three scientists, one Hugo de Veris, another Lorentz, and third von Tesma. All these three scientists rediscovered his work. These three scientists, Hugo de Veris, Lorentz, and von Tesma, all these three scientists rediscovered the work of Mendel. Mendel's work and परत rediscover केला. All these observe cell division, because at this time the advancement in microscopy. There is a microscope is developed. Under the microscope, we observe cell division and structure of cell, structure of chromosome. मायक्रोस्कोप खाली त्यांनी ऑब्झर्व केलं, एक सेल डिव्हिजनचं स्टडी केलं त्यांनी, सेल कशी डिवाइड होतात ते त्यांना दिसलं मायक्रोस्कोप खाली आणि स्ट्रक्चर दिसलं क्रोमोझोमचं. तर हे या थिंग्स इथे त्यांनी ऑब्झर्व केल्या या तिन्ही सायंटिस्टने. आणि नेक्स्ट आहे ते म्हणजे सुटॉन आणि बॉबेली. या द नेक्स्ट सायंटिस्ट वॉल्टर सुटॉन अँड थिओडोर बॉबेली. दिस सुटॉन अँड बॉबेली both have a landmark in the genetics. These two scientists in 1903 studied parallel behavior of Mendel's factor and behavior of chromosome. Other scientists does not accept his work, but De Vries and other two are that rediscovered his work. Then these two, Sutton and Boveri, in 1903 studied parallel behavior of this Mendel's factor or gene and behavior of chromosome. They study how genes and how chromosomes are behaved in meiosis. These two studies are followed in the meiosis. Then these two scientists, Sutton and Boveri, put forth a theory called chromosomal theory of inheritance. These two scientists put forth this theory called as chromosomal theory of inheritance. This theory identifies chromosome as the carrier of genetic material. या दोघांनी ही theory put forth केली, जिचे नाव आहे chromosomal theory of inheritance. आणि यांनी सांगितलं, यांना आयडेंटिफाय झालं की क्रोमोझोम ॲज अ कॅरियर ऑफ जेनेटिक मटेरियल. म्हणजे जे काही जेनेटिक मटेरियल पॅरेंट्सकडून ऑफस्प्रिंगला दिलं जातं ते कशावर दिलं जातं नेमकं, कोण कॅरी करतं त्याला पॅरेंटकडून ऑफस्प्रिंगकडे, तर हे जेनेटिक मटेरियल कॅरी होतं ते म्हणजे क्रोमोझोम्सवर. हे पहिल्यांदा कोणी सांगितलं, ह्या सुटॉन आणि बॉवेरी त्यांच्या थेअरीमध्ये. त्या थेअरीचं नाव आहे क्रोमोझोमल थेअरी ऑफ inheritance. This theory states number of things, number of statements, assignment. First, this theory states that chromosomes are always in a pairs in a eukaryotic cell. In prokaryotic cell there is a single chromosome, there is no homologous chromosome, but in eukaryotic cell the chromosomes are always in a pair, 2 or 4 or these are not in odd number, they are always in a even number. All chromosomes are present in a pair. First number chromosome coming from mother and other is from father. Second, third, these are always in a pair in a eukaryotic cells. These during this gamete formation, homologous chromosomes pair, segregate and assort independently. Means these chromosomes at the time of meiosis cell division you studied in 11th standard. In this meiosis is nothing but the reduction division. When there is a 46 chromosome, this division, in this division 23 in one gamete and 23 in other gamete. Means 23 chromosomes are divided. At the time of meiosis, these chromosomes 1 number, 2 number, they forms the pair during meiosis, and not only form the pair, they separate, they segregate from each other. The one to the one pole, another to the another pole. This one chromosome goes to one pole, another is another pole. Means these are goes in a separate two daughter cell, two gamete cells. They are separated and assort independently. They are not mix each other. They assort, they separate independently. The gametes contain only one chromosome of a pair. आता साहजिक आहे gametes हे जी प्रोसेस आहे फॉर्मेशन ऑफ gametes ही meiosis division ने होते. Meiosis division, reduction division आताच पाहिलं आपण. Reduction division means 46 क्रोमोझोम असतील तर 23 एका गॅमेटमध्ये, 23 दुसऱ्या गॅमेटमध्ये जाते. जर 23 म्हणजे एका पेअर मधला एकच क्रोमोझोम खेचून एका पोलला नेला जातो, एका गॅमेट मध्ये आणला जातो, म्हणजे वन गॅमेट कंटेन ओनली वन क्रोमोझोम ऑफ अ पेअर. म्हणजे एका पेअरचा एकच क्रोमोझोम असतो एका गॅमेट मध्ये. देन न्यूक्लियस ऑफ दिस गॅमेट. आता ही जी गॅमेट तयार झाली, जी सेल तयार झाली, जर मेल गॅमेट असेल तर स्पर्म आणि फिमेल गॅमेट असेल तर एग. तर या एग आणि स्पर्मच्या न्यूक्लियस मध्ये जे क्रोमोझोम्स आहेत, हा एग आहे आणि हा स्पर्म आहे, या दोघांच्या न्यूक्लियस मध्ये ऑल हेरिडिटरी कॅरेक्टर क्रोमोझोम्स असतात, जे काय कॅरी करतात, ऑल हेरिडिटरी कॅरेक्टर्स. आपल्याला पॅरेंट कडून फक्त काय मिळत आहे, तर हे गॅमेट्स मिळतात. मेल पॅरेंट कडून स्पर्म आणि फिमेल पॅरेंट कडून एग्स. आणि याच्यामध्ये असणारे जे न्यूक्लियस आहे ते मिक्स होतात, त्यांचे क्रोमोझोम्स मिक्स होतात. म्हणजे न्यूक्लियस मध्ये जे क्रोमोझोम्स आहेत त्यांच्यावर ऑल हेरिडिटरी कॅरेक्टर कॅरी केले जातात. सगळे कॅरेक्टर्स त्याच्यावर कॅरी होतात. मेल गॅमेट स्पर्म अँड फिमेल गॅमेट ओव्हा और एग. मेल गॅमेटला काय म्हणतो आपण, स्पर्म, आणि फिमेल गॅमेटला काय म्हणतो, एग और ओव्हा. दीज कॅरी ऑल हेरिडिटरी कॅरेक्टर. आपल्याला पॅरेंट कडून काहीच नाही मिळालं, फक्त हे दोन गॅमेट्स मिळाले. म्हणजे गॅमेट्स मध्येच काय कॅरी केले जात आहे, ऑल कॅरेक्टर्स. These gametes is nothing but the link between. These are gametes, male gamete, female gamete. These gametes are the link between parents and offspring. म्हणजे दोघांना जोडणारा दुवा काय असेल तर पॅरेंट आणि ऑफस्प्रिंगला एकमेकांना जोडायचं काम कोण करतात, की गॅमेट्स. गॅमेट्स पॅरेंट कडून आले, फ्यूज झाले आणि मग ऑफस्प्रिंग तयार झाले. म्हणजे हे दोघांच्या मधली काय आहे, लिंक. जशी प्रत्येकाच्या लाईफ सायकल मध्ये मियोसिस डिव्हिजन इम्पॉर्टंट आहे. आता पाहिलं आपण फॉर्मेशन ऑफ गॅमेट्स, मियोसिस डिव्हिजन ने कंप्लीट होता. 23 डिवाइड होतात क्रोमोझोम, हाफ सेट्स एकामध्ये दिले जातात. म्हणजे हाफ या हाफ सेट त्याच्यामध्ये हॅप्लॉइड गॅमेट आहे. हा एग्स कसा आहे, या नंबर, आणि या स्पर्म मध्ये सुद्धा किती क्रोमोझोम्स आहेत, या नंबर. म्हणजे 23 क्रोमोझोम आहेत, याच्यामध्ये 23 क्रोमोझोम्स आहेत. फक्त 23 क्रोमोझोमने होल बॉडी तयार करू शकत नाही. म्हणजे फक्त एग्स ऑफस्प्रिंग तयार करू शकत नाही किंवा फक्त स्पर्म ऑफस्प्रिंग तयार करू शकत नाही. मग दोघांचे जे क्रोमोझोम्स आहेत ते मिक्स होणे, दोघांचे रिस्टोर होणे, ज्याला फ्युजन, पुढची स्टेप फर्टिलायझेशन. Here we will start the new topic of inheritance and variation. Third chapter, inheritance and variation. The next bit is chromosome. Chromosome, this term coined by W. Valier in 1888. This term given by, this is coined by, this chromosome, this word is coined by Valier in 1888. What is the meaning of this chromosome? Chromo means color and zoma means body. Means colored body seen in the eukaryotic cell called as chromosomes. When we stain the cell and see under microscope, then colored body seen under the microscope called as chromosomes. In cell, eukaryotic cell, there is a filamentous structure found in the nucleus. This is the cell wall, nuclear membrane, nucleus, nucleolus, and this is the filamentous structure found in the nucleus. This filamentous body is called as chromosomes. Filamentous bodies in eukaryotic cell called as chromosome. Then we will see the detailed structure of chromosome, size, what is the chemical nature of chromosome, what is the function of chromosome, what is the number of chromosome, what is the actual structure of chromosome. Here first we will see the size of chromosome. The size of chromosome is 0.1 to 33 micrometer in length and 0.2 to 2 micrometer in thickness. म्हणजे जी लांबी असते क्रोमोझोमची या रेंज मध्ये असते 0.1 to 33 मायक्रोमीटर या रेंज मध्ये आणि 0.2 to 2 मायक्रोमीटर या रेंज मध्ये थिकनेस असते. They are variable in size in variable cells. They are visible during only cell division. Why they are visible only during cell division? Because at the time of cell division, packaging of chromosome takes place, condensation of chromosome takes place. So they seen only during cell division. They have capacity, they capable of self-replication. Self-replication, they duplicate themselves, they replicate themselves, they synthesize themselves. हे जे chromosomes आहेत हे DNA पासून तयार झाले and DNA have the capacity to replicate themselves or self-replication. And what is the role of this chromosome? They play a vital role, important role in heredity, mutation, variation and evolution. Heredity, they carry or transfer character from one generation to the next generation. Sutton and Boveri state that the chromosome is the carrier of genetic material. Means genetic material or characters carried by on a chromosome, means heredity. What is mutation? Genes suddenly change their location and their characters change, called as mutation. What is variation? During the crossing over, the segment of homologous chromosome between non-sister chromatid transferred, and this is called as crossing over. This change the character, location change the character, called as variation. And what is evolution? Evolution means developmentary changes in the species, in the eukaryotic species. New species of eukaryotic organism are evolved. This evolution, basic of evolution is the variation. And what is the tool of evolution? Is the DNA or chromosomes. What is the chemical nature of chromosome? Chromosomes are made up of DNA, histone and non-histone protein. Chromosome made up of histone and non-histone protein. It is a packaging material and main material is the DNA. Number of DNA, number of chromosomes, and they are packaged on histone and non-histone proteins. Means chemical nature of chromosome is DNA, histone and non-histone proteins. What is the function of chromosome? We are already see in the Sutton and Boveri. They are carrier of heredity. Carrier. They carry characters from one generation to next, means they are a carrier of heredity. Then what is the number? Number of chromosome is specific and constant for a particular species. For example, in human being, 46 chromosome. This number is constant. It is not for change to Indian people and Australian people. It is common. It is stable and it is specific. For example, in honeybee, there is a 16 in male and 32 in female. This is the specific number and constant number. In birds, 46 chromosome. Then what is euploid? Ploidy, euploidy and uneuploidy. These are the terms according to number. What is ploidy? Ploidy means a degree of repetition of primary basic number of chromosome. Primary basic number of chromosome is X. And X repetition. How many times you repeat? X, 2X, 3X means how many times? Degree of repetition of primary basic number of chromosome, that is X, called as ploidy. And what is euploidy? Euploidy means chromosomes number in a cell is the exact multiple of primary basic number. What is euploidy? Chromosome number in a cell is the exact multiple of primary basic number. Primary basic number of chromosome means X is equal to N, then N into N, 2N, 3N, 4N means multiple. Exact multiple of primary basic number is the euploid. How many times they multiply? Or it is the chromosome in a cell is the exact multiple of primary basic number. It includes number of terms like monoploids or haploids. Monoploids or haploids means what? The organism with one set of chromosome in a cell. Only one set, X is equal to N. One set of chromosome in a cell called as haploids. What is diploids? Cell with two sets of chromosomes, 2n number, two sets of chromosome called as diploid. Cells with three sets of chromosome called as triploids. Cells with four sets of chromosomes, 4n is the tetraploid or so on, pentaploid, hexaploid, etc. Then what is the uneuploid? Uneuploid, it is opposite term to the euploid. Euploidy, uneuploidy. What is the definition of euploidy? Chromosome number in a cell is the exact multiple of primary basic number. Here only chromosome number in a cell is not the exact multiple of primary basic number. म्हणजे जो नंबर असेल आपल्या पॅरेंट्स कडून ऑफस्प्रिंग मध्ये जो क्रोमोझोम नंबर आहे ते फिक्स असायला पाहिजे. फॉर एक्झाम्पल पॅरेंट मध्ये 46 आहेत ह्युमन बीइंगच्या, तर त्याच्या चाइल्ड मध्ये ऑफस्प्रिंग मध्ये सुद्धा किती असायला पाहिजे 46. परंतु जर हे 46 नसतील ते ऑफस्प्रिंग मध्ये तर चेंज असेल नंबर, तिथे एखाद्या क्रोमोझोमचं ॲडिशन झालं असेल किंवा एखाद्या क्रोमोझोमचं डिलिशन झालं असेल, दिस इज कॉल्ड ॲज अनयुप्लॉइडी. means not exact multiple of primary basic number. Addition of chromosome or deletion of chromosome. Addition of chromosome, for example, in human being, when there is a 47 chromosome, 21 trisomy, it is a Down syndrome. When one chromosome extra, there is a number of symptoms, and this syndrome is called as Down syndrome. Or Klinefelter syndrome, 47 chromosomes. Or deletion means less number, Turner syndrome, 45 chromosomes, not 46, one chromosome is less, 45 chromosomes, this is a Turner syndrome. All syndromes are seen in a next bit here. These are the example of Turner syndrome or Down syndrome, Klinefelter syndrome. These are the example of aneuploidy. Means chromosome number in a cell is not exact multiple of primary basic number. म्हणजे जे पॅरेंट मध्ये ते मुलांमध्ये तेवढे नसतात, एक तर कमी असतील किंवा जास्त असतील, याला म्हणायचं अनयुप्लॉइडी.